process of Xenophus levis. So what you are looking at are the different schematic diagrams of the gastrulation process of Xenophus levis. This is the blastula of the Xenophus levis with a characteristic blastocele over here. So you see a blastocele over here. So this is a blastocele that is present in the blastula stage, and the gastrulations will begin somewhere over here, and this region is called the marginal zone, because this is the margin between, or this is the margin between the animal pole and the vegetal pole over here. So this region is called the marginal zone, and this is the equator of the egg, and the gastrulation begins somewhere over this region, somewhere just little bit below the equator regions at the marginal zone. The gastrulation begins with the formation of the characteristic bottle-shaped cells like this. So these cells are formed first at the marginal zone. These cells are called the bottle cells, as is shown here. So these bottle cells are shown here, and with the formation of the bottle cells, the gastrulation begins. The gastrulation involves the different types of cellular movements in the embryo. Here you can see the bottle cells are formed here. The bottle cells are formed in this region, and after the formations of the bottle cells, the cells start to migrate into the interior of the embryo. Okay, so this is the, if this is the embryo, this is the dorsal lip of the blastopore, and the cells are migrating into the interior of the embryo. Cells are rolling into the interior of the embryo. This process is called the involution. The process by whereby the cells migrate into the interior of the embryo along the roof of the blastocells. So this is the blastocells, and the cells are migrating along this roof of the blastocells into the interior of the embryo, and this process is called the involution. And this region is the dorsal lip of the blastopore. Actually, it is a lip that is formed at the dorsal side. It is the dorsal side and it is the ventral side, and the lip is formed at the dorsal side. It is called the dorsal lip of the blastopore. Okay, so now if you look at this diagram, you see that a new cavity is formed that is called the Archenteron, and as this Archenteron continues to form, the blastocele is getting displaced, and it is beautifully shown here. The blastocele is getting displaced and the Archenteron cavity expands. This is the dorsal lip of the blastopore, and as this process continues, so you have another lip that is called the ventral lip, and this is the remaining endoderm. So this is endoderm, this is endoderm, this red cell layers are the mesoderms, and the surface layer, the blue cells, are ectoderms. The ectoderms are shown in the blue cells. These blue cells are migrating in this direction as is shown by the arrow. So actually it is just migrating in this direction and also these directions, just to enclose the entire embryo, and this process is called the epiboly. So one group of the cells are moving like this, another group of the cells are moving like this. So this is called the epiboly, and this is called involution. Okay, so after the certain times, as after the certain time, the as this cavity continues to form, so you have a remaining patch of the endoderm that is called the yolk plaque. Now look at this diagram. So you see a ventral leaf, you see the dorsal leaf. This is involuting cells, and this, as these cells are involuting through this dorsal lip of the blastopore, this region is called the IMZ, or involuting marginal zone. And as this continues to, as this cavity continues to form, the blastocyst is getting displaced and eventually the blastocyst disappears. Now you are left with this Archenteron, and you might have heard about the name Deuterostome. The Deuterostomes are those animals in which the blastopore develops into the anus, and the anus develops first and the mouth forms second, and this is the blastopore or the dorsal lip of the blastopore that is going to develop into an anus. Okay, so this is the characteristic. Actually, in this picture the characteristic cellular movement is shown. This is the surface ectoderm shown in the blue, and this is the endoderm, and this is the pharyngeal endoderm. Okay, so the bottle cells are formed here. As the bottle cells are formed, the involution starts, the cells start rolling inside the interior of the embryo. Osteoid cells actually are traveling along the roof of the blastocells, and the first group of the cells that will involute through this interior of the embryo are the pharyngeal endoderm cells. The next group of the cells involuting through this dorsal lip are these light color red cells over this region. So these cells, so this group of the cells, sorry, so this group of the cells. Okay, so and then these red deep colored cells shown here actually eventually migrate into the interior of the embryo through this dorsal lip of the blastopore, and they are the precursor of the cord mesoderms that will give rise to the notochord. And notochord is very important in inducing the formation of the axis, embryonic axis. So embryonic axis is first specified by the development of the notochord, and this notochord is derived from this group of the mesodermal cells called the cord mesoderms. Now the question is, so you have, this is my embryo, this is my blastula, the dorsal, this is the marginal zone, let's say this is the marginal zone. So dorsal lip of the blastopore will form somewhere over here, and the cells will migrate into the interior of the embryo. The ectoderms will uh, cover the entire embryo in a process called epiboly, and this involution movement leads to the formations of what is called the Archenteron. So this Archenteron, and leading to the displacement of blastocells, and the cells involuting through these cells in actually, okay, so a group of cells involuting through this dorsal lip of the blastopore. So you have, as I have shown before, that will form the notochord. Okay, so they will form the notochord. Okay, so and notochord is specifying the anterior posterior axis. So notochord is specifying this anterior posterior axis. Now the question one could ask, the what are the specialized property of the dorsal lip of the blastopore? Okay, so what would have happened? So one could ask that what would have happened if we had another dorsal lip of the blastopore over here? So would we have another notochord? So let us see with this experiment carried out by the Spemann and Mangold. The Spemann and Mangold take two differentially colored newt embryo. Okay, so these are the two differentially colored embryo. These are the normal dorsal lip of the blastopore, and the cells are involuting into the interior of the embryo through this dorsal lip of the blastopore and forms the notochord. Okay, so basically it is normally happening, and these are the surface ectoderm. Okay, so as I have shown that these ectoderms will cover the entire embryo, and this is the surface ectoderm, and it is fated to become this epidermis. Okay, now what the Spemann and Mangold did, so they took this dorsal lip of the blastopores from a donor embryo and transplanted it into a recipient embryo in a region that is fated to become the epidermis. And now they observed what would happen, and to their surprise they found another dorsal lip of the blastopores where they have implanted the donor tissue, and the cells are involuting into the interior of the embryo through this dorsal lip of the blastopore, leading to the formation of another notochord. So this is the normal notochord formed from this dorsal lip of the blastopore, and this is my another notochord formed from this dorsal lip of the blastopore, from this dorsal lip of the blastopore. Okay, so actually, so this embryo is supposed to have one notochord, but instead of having one notochord, it is now having two notochord. Okay, just because it has another dorsal lip of the blastopore being implanted over here. Okay, so when this embryo developed, it would contain the two axis and the two head. So this is called the conjoined twin. That means the dorsal lip of the blastopore. So this experiment is basically providing some significant clue about the property of the dorsal lip of the blastopores. So we would say the dorsal lip of the blastopore is the organizer that organizes the embryo into an anterior posterior axis that defines the axis. So that's why it has a special property wherever you implant it. So if you implanted this dorsal lip of the blastopore over here, if you implanted the blastopore over here, it will direct the formation of the notochord wherever you have implanted. That means the cells constituting this dorsal lip of the blastopores are special. But what are so special about these cells? Now look at this image. This is the animal capsules fated to become the ectoderm. This is the marginal cells that will involute through the interior of the embryo and becomes the mesoderm. The endoderm will also involute through this dorsal lip of the blastopores. This is the vegetal pole, and the cells of this vegetal pole actually become the endoderm. Okay, so let's say we obliterated this blastocell. So we obliterated these blastocells and stitched these animal capsules or placed these animal capsules over these endodermal cells. Now you observe that this group of the cells, so this is basically the animal capsule. So these cells are animal capsules that are fated to become the ectoderm, but instead of developing into the ectoderm, it is now following the mesodermal fate. When it is placed just above when it is sitting just above these endodermal cells, so it is becoming the mesoderm. It gives rise to the mesoderms. So why it is giving rise to the mesoderm instead of ectoderms? And the scientist, it turns out to be that this group of cells secrete some diffusible factors, some diffusible proteins, and those diffusible protein direct the formation of the mesoderms. So in this case, these diffusible factors come in contact with the marginal cells and induce the formations of mesoderms. When these diffusible factors come in, and this Archenteron prevented this diffusible factor from reaching up here. But when this Archenteron is obliterated, the diffusible factors would come in contact with these animal capsules that is supposed to develop into the ectoderm. But now, in the influence of this diffusible factor, it is becoming an ectoderm. And then Okay, so basically these group of cells are very important. So the dorsalmost vegetal cells, so these are the dorsalmost vegetal cells. So this is the dorsal region, this is the ventral region, and this is the vegetal pole, and this is animal pole. Okay, so this is the dorsalmost vegetal cells. So this dorsalmost vegetal cell is secreting something that gives this dorsal lip of the blastopore its organizer property. Because of this group of cells, the dorsal lip of the blastopore is able to direct the axis formation. If we remove this group of the cells, then it would not be possible for the organizer or for the dorsal lip of the blastopore to organize the embryonic axis, and that's why these cells are called the organizers, and these dorsalmost vegetal cells are called the Nieuwkoop center, named after its discoverer. Okay, so basically the Nieuwkoop centers are these dorsalmost vegetal cells that is releasing some diffusible factor, which in turn induce the dorsal lip of the blastopore to form the mesoderms. So in if we place this animal capsules that is fated to become the ectoderm in contact with this Nieuwkoop center, then it will eventually become the mesoderm forming tissue. It will eventually become the tissue can can have the ability to direct the formations of embryonic axis. So the next questions that one should ask is, what are the molecules, what are the diffusible factors that gives this Nieuwkoop center, that endows the Nieuwkoop center with the ability to induce the development or induce the formation of organizer. Okay, so what are the diffusible factors? This diffusible factor is the beta-catenin. The diffusible factor is beta-catenin. Let me draw. This is the dorsal region, this is the ventral region, this is my new Nieuwkoop center, and this is the dorsal lip of the blastopore, and this is going to be my organizer. Okay, so I am denoting it as O, and this is Nieuwkoop center, and that's why I am denoting as N. Okay, so and this is the, let's say this is the cavity. Okay, now it is secreting something that is inducing these cells to become the organizer. Okay, so beta-catenin messenger RNA. So beta-catenin is a secreted molecule. So beta-catenin messenger RNA is located throughout the embryo, but beta-catenin is protein is only present in this dorsal site. Okay, so if it is the beta-catenin, then this beta-catenin is localized at the dorsal side. It is not present in the ventral side. So how does this possible? The messenger RNA of beta-catenin, so beta-catenin messenger RNA are present throughout the embryo, but only it can be translated into the, only so you can find the protein, beta-catenin protein, in the dorsal region of the embryo. So how does the beta-catenin is stabilized at the dorsal region of the embryo, and it's and it's stabilized? So the it is localized at the dorsal region of the embryo because it is stabilized here. It is not present in the ventral region of the embryo because it is degraded from the ventral region. Now how does this happen? Actually, I will discuss this. I will discuss the exact mechanism of the localizations of the beta-catenin at the dorsal region of the embryo. How does the beta-catenin accumulate at the dorsal region of the embryo, not the ventral region of the embryo, in the next video.